MCAT 2017 Cram, Biochemical and Biological Foundations of Living Systems. Passage 10, glucose-6-phosphate dehydrogenase, and the most important function of the pentose phosphate pathway. All right. Paragraph 1. The pentose phosphate pathway plays an important role in the synthesis of lipids and nucleotides. It also helps cells produce antioxidants that detoxify hydrogen peroxide, a main source of oxidative stress in animal cells. Figure 1. The pentose phosphate pathway converts glucose 6-phosphate to ribulose 5-phosphate and NADPH. Okay. Paragraph 3. In the case of the experiment with G6PD null cells, mean cloning efficiency was 5.7% plus or minus 1.8%. Mean cloning efficiency for control cells was 51.2%. Whoa, what a difference. Plus or minus 9.4%, but a wide range or standard deviation. These results are depicted in Figure 2. Another cloning efficiency assay was performed under culture conditions in which oxygen concentration was reduced by 40%. The results of this experiment are depicted in Figure 2. Figure 2. Mean cloning efficiency for G6PD null cells versus control in 24% oxygen and 13% oxygen. Which of the following hypotheses best explains the low cloning efficiency of G6PD null cells in standard cell culture conditions? That would be 24% oxygen. Is it going to be A, absence of the rate-limiting enzyme of the pentose phosphate pathway prevents synthesis of nucleotides needed for cell division? Is it going to be B, absence of the rate-limiting enzyme of the pentose phosphate pathway prevents production of electron acceptor molecules? Or is it C, absence of the rate-limiting enzyme of the pentose phosphate pathway sensitizes cells to harmful oxidative stress? I'll give you a moment to decide. All right. Okay, so a 5.7% cloning efficiency in 24% oxygen suggests that G6PD null cells are at a survival disadvantage compared to the control cells with functional G6PD, okay? So if disruption of nucleotide or lipid synthesis was causing the survival disadvantage for G6PD null cells, one would not expect a decreased oxygen concentration to improve cloning efficiency, okay? So cloning efficiency for G6PD null cells is comparable to cloning efficiency for control cells when cultured in only 13% oxygen. This suggests that the absence of the rate-limiting enzyme of the pentose phosphate pathway, sensitizes cells to harmful oxidative stress, in the form of hydrogen peroxide. So when there's less O2 present, there's less oxidation going on, okay? That's why lowering the oxygen content actually helped. Although we would expect it to hurt, it did the opposite. Transketolase and transaldolase are enzymes that catalyze the conversion of ribulose 5-phosphate into intermediaries of the glycolytic pathway. The cloning efficiency of G6PD null cells in 13% oxygen could be explained by reactions catalyzed by these enzymes having which of the following properties under the physiologic conditions seen in this experiment? Is it going to be A, a delta G much less than 0, B, a low K catalysis, C, a delta G close to 0, or D, a high Km, K sub M rather, or Michaelis-Menten constant? I'll give you a moment to think. All right. Okay, so in order to answer this question, you definitely had to know something about reaction kinetics outside of the passage, or you could have used another thought process based on the passage to arrive at the answer choice, but I'm not sure what that would be, okay? So reducing the oxygen content from here to here restores the cloning efficiency of the G6PD null cells to levels seen with control cells, okay? Which implies that nucleotide synthesis from ribulose 5-phosphate proceeds in the absence of the G6PD enzyme, okay? Something you would have to know is that reactions with delta G's close to 0, they're reversible. They don't just proceed in one direction, okay? So if it were that the reactions catalyzed by transketolase and transaldolase are reversible under biologic conditions, then ribulose 5-phosphate can be produced from glycolytic intermediaries, all right? And hence, nucleotide synthesis can occur. So the cloning efficiency of G6PD null cells in 13% oxygen could be explained by the reaction catalyzed by these enzymes having a delta G close to 0. All right, okay. Okay, so NADPH is also produced by the enzymatic reactions in the mitochondria. Based on this fact, which of the following cell types would be most susceptible to oxidative stress, assuming G6PD deficiency? Is it A, neurons, B, hepatocytes, which are liver cells, C, red blood cells, or D, muscle cells? I'll give you a moment to think. Press pause if you need to. Okay, so mitochondria are membrane-bound organelles where much of cellular respiration occurs. Red blood cells are unique amongst animal cells because they don't contain the normal complement of organelles. Vertebrate red blood cells contain no organelles whatsoever, while other animal cells or RBCs may only contain a nucleus, okay? So they lack mitochondria. A cell which contains neither mitochondria nor G6PD would have very limited NADPH producing abilities, and thus would be extremely susceptible to oxidative stress Hence, red blood cells with G6PD deficiency would be most susceptible to oxidative stress. All right? Okay. G6PD is known to be subject to inhibition. Okay, so this enzyme would act somewhere along here, between here and there, maybe about right here, in the pentose phosphate pathway. Given the metabolic role of the pentose phosphate pathway, that is what it produces, which of the following is least likely to be an inhibitor of G6PD? Would it be A, NADPH, B, epinephrine, which is also known as adrenaline, the adrenaline hormone? Would it be C, insulin? Or would it be D, ribulose 5-phosphate? I'll give you a moment to think. All right. Okay, so the pentose phosphate pathway produces molecules that are important for the biosynthetic activity of cells, such as nucleotides, lipids, and antioxidants, okay? The products of metabolic pathways commonly inhibit the further activity of the pathway in question. For instance, NADPH would inhibit the G6PD enzyme, which isn't depicted, but it works somewhere over here, which would in turn downregulate the pentose phosphate pathway. Okay, so things would start to slow down. Insulin, however, in addition to its function in the transport of glucose, it sets off a metabolic mechanism whereby the GLUT4 receptor intakes the glucose molecules into the cell. It also has a more general function of downregulating catabolic pathways, that is, pathways that release energy, and upregulating anabolic pathways, pathways that store energy and synthesize molecule. Hence, insulin is least likely to be an inhibitor of the gene G6PD, okay? Not the gene, the enzyme. All right. According to Figure 1, ribulose 5-phosphate is important for the synthesis of nucleotides, as shown here in the pentose phosphate pathway. Which of the following molecules contain nucleotides and thus would be expected to be depleted in a cell with low levels of ribulose 5-phosphate production? Would it be 1, ATP, which is short for adenosine triphosphate? Would it be 2, NADH, which stands for nicotine adenine dinucleotide? One nucleotide here, one nucleotide there. Or would it be 3, TNA, which is short for deoxyribonucleic acid? I think I pretty much gave away the answers. So you decide, is it going to be A, 1 only, B, 3 only, C, 1 and 3, or D, 1, 2, and 3? You decide. I'll give you a moment to think. This should be really straightforward. All right. Okay, so nucleotides basically have 3 requirements. The first one is a nitrogenous base. we have two pictured here, a 5-carbon sugar, which would be ribose, and one or more phosphate groups, okay? So this is NADH, which is basically the reduced form of nicotine adenine dinucleotide, and this is the N that's reduced. It has a hydrogen or a hydride if you want to look at it in that way. So yeah, this is basically a nucleotide, all right? Adenosine triphosphate consists of adenosine and adenine nucleotide attached via a glycosidic bond to the 5-carbon sugar ribose, and it's bonded to 3 phosphate molecules, the ribose that is, okay? So yeah, this is a nucleotide as well. And last but not least, we have our DNA, deoxyribonucleic acid, which consists of repeating sequences of nucleotides attached via, one chain. One anti-parallel chain is connected via the sugar phosphate backbone, and then anti-parallel strands are connected through hydrogen bonding of nitrogenous bases, okay? Yeah, so basically ATP, NADH, and DNA all have nucleotides. So a cell with low levels of ribulose 5-phosphate production would also likely have low levels of ATP, NADH, and DNA as well. This could be very problematic. All right, okay.